Hi, good afternoon. We're going to get started. My name is Monica Avila, and I'll be the moderator of this session. I've heard some really good talks today, so hopefully our crew will continue the trend and give you some new and interesting information. The title of our session is Cord Blood as a Platform for Technology. So we're going to be talking about different ways that we use cord blood aside from transplant. The three of us will speak, and then we'll have a question and answer session at the end of all of our speakers. So I'm going to go ahead and get us started. Again, my name is Monica Avila, and I work at Selencos, which is a company in Houston. I have been there for a little over 4.5 years. I started my career at MD Anderson Cancer Center in their stem cell transplant laboratory, where I worked on several cord blood expansion protocols and phase 1 clinical studies there. I then moved on to Cord Blood Bank, and I got to see the other side and work at the Cord Blood Bank there in the manufacturing area. So I've been very fortunate to be able to see both sides of the coin, and little did I know that was going to actually come in very handy with my work here at Selencos. I have nothing to disclose. So just a little bit about us. Selencos, again, is a small company located in Houston, Texas. The technology originated at MD Anderson Cancer Center in Dr. Simrit Parmar's laboratory. In 2016, she was able to secure sufficient funding to transfer our technology out of MD Anderson and start up Selencos. Our team consists of 12 full-time employees, and we have various consultants that we work closely with. As you can see, we're a very small team, but we are mighty. We currently hold 4 INDs. We just submitted our 5th IND, and that's undergoing FDA review as we speak. Actually, we're still answering questions right now. And we also have been a part of 11 compassionate INDs that we've helped file. We have our own GMP facility on site, and we do our own manufacturing. And we also serve as our drug depot, so we also are in charge of distributing our IP to the clinical sites. Just a little bit of our vision. So Selencos' vision is to discover global cell therapeutics for treatment of autoimmune diseases and inflammatory conditions. And our mission is to create widespread application of cord blood regulatory T-cell therapeutics. So just in line with our mission, our vision, which is to treat autoimmune and inflammatory conditions, we decided as a company to really focus our efforts on indications where there really is not much out there as far as therapy options. And one of these is, I'm listing them here, so we have acute respiratory distress syndrome, ARDS. So this is the inflammation in the lungs that's as a response to pulmonary damage. This was front and center during the COVID epidemic, where we saw a lot of individuals who fell ill, had this syndrome, and really led to intubation. And you know, unfortunately, once you're intubated, the chances of mortality greatly increased. Myelofibrosis is another indication that we're treating, and this is a cytokine and chemokine-driven inflammatory disease in the bone marrow. There are some treatments out there, drugs that are out there to treat this successfully, but there's still a portion of the population that have a suboptimal response. And some of the hypothesis behind why the drug isn't working is that there's just really severe inflammation in the marrow that prevents the drug from coming in and doing its job. So we are going to work in tandem with the company to give a infusion of our product to help calm the inflammation in the marrow, and then they will proceed to give their drug, and our hope is that we'll have a better prognosis. ALS, everyone knows that. That's something that's has been on our sites since Solenco started. This is a neurodegenerative disease, which really no therapies exist at the moment. There are some drugs that are coming out with minimal benefits, and it's really important to us because it's such a rapid decline when patients start deteriorating. It's at a really rapid pace. Mortality is really sometime from 2 to 3 years from onset of symptoms. So it's something that we are working hard to help. Our weapon of choice is T regulatory cells. So this is a specialized subpopulation of T cells that are formed in the marrow, and then they move out to the thymus, where they're differentiated or pushed out into the peripheral blood as helper cells, waiting for to be called to duty. The main role of T regulatory cells is to maintain homeostasis of the immune system. So if we can imagine whenever there's an infection or some kind of injury, you have this rush of cells, immune cells, to the area. So you have your first responders that come, and then they do cytokines and signaling, recruit other forces that come in. And if you can imagine a battlefield, there's a lot of destruction. And so this is what's happening in that area. So you have inflammation, you have a lot of destruction. So the Tregs come in, and their job is to suppress, to calm everybody down, to move everybody away from the area so that the healing can begin. So Tregs suppress by numerous different ways. And one of this is to secrete anti-inflammatory cytokines. IL-10 is an example of this. So Tregs come in, they secrete IL-10. This binds to the macrophages and really is like an on-off switch, so it turns them off and kind of shuts them off, and they move out of the area. They also secrete TGF-beta, which is a kind of a call for more Tregs to come. So it kind of brings in the reinforcements to come and help out. They also are capable of cell lysis, so they can actually bind to certain cells and secrete enzymes that will kind of just chew them up and destroy them and get them out of the way, immune cells. They also block immune signaling. So IL-2, cytotoxic T cells use this to get activated. So T regulatory cells have a lot of receptors, IL-2 receptors on their surface, and they have a higher affinity for this IL-2. So they come in and work like a sponge that comes in and kind of sucks up that IL-2 and keeps it from activating other cells. This is a different slide here, but they also bind to APC cells, and this keeps them from going around and activating other immune cells. So this is just a diagram of Treg function. So as we discussed, there's cytokine production that helps, they secrete anti-inflammatory cytokines. There's also the cell-to-cell regulation, so they bind to APCs and keep them from going and talking to other cells and getting them going. Then you have your granzyme production, where they kill the NK cells and get them out of the way. And then you have your IL-2 competition, where the Tregs, soak up the IL-2 and get them out of the picture. Sorry, this is not my slideshow. So just, there's a lot of benefits of using the Treg cells. So using cord blood as our starting material. One of these is that they're rich in stem cells. This is something that I think all of us know. So this has helped us when we're expanding T regulatory cells. So they have a higher fold expansion since they come from immature cells. There's also low immunogenicity, which allows for a greater HLA mix match. But we have also taken it a little bit further and joined other groups who are creating off-shelf products. So we completed a clinical trial where we gave products from cord bloods with no HLA matching at all. And we had no SAEs from those. There's also the low risk of infectious disease transmission. We're all aware of the donor screening and testing process here, so we feel fairly confident that what we're giving is not hurting others. And also the surplus inventory. I think I've heard a lot today about how banks have a lot of products in their inventory, how there's a decline in cord blood transplants. So what we've been able to do is kind of leverage that and work with different cord blood banks so that we can help to secure secure a supply of cord bloods for us to use for our manufacturing, and also help them to recoup some of their costs. I know it's very expensive, so all costs won't be recouped, but at least they can recoup some. But more importantly, to move some of these units that otherwise really wouldn't be suitable for transplant, move those units out of their inventory so that they can have space for maybe a higher quality cord. So right now, some of the current therapies are used for Tregs, use autologous donors. So this slide here is really just talking about the comparison between autologous and cord. We talked about matching, how cord blood can be a universal donor to patients. Source, patients for autologous have to go through an apheresis process, and as manufacturers, we have to wait for that process to take place. With cords, we already have a cryopreserved unit. Stability, there have been studies that show that mature T regulatory cells they have the capability to actually differentiate back into cytotoxic cells, which is something that we don't want in an inflammatory situation. Tregs, we've been able to demonstrate stay stable, so they keep their phenotype throughout the process until they're cleared from the body. Scale, so for autologous, you're really producing product for one patient, where for cord blood, we're able to manufacture multiple products and treat multiple patients from one cord blood. Consistency, we are very lucky. We take our cords from accredited cord blood banks here in the United States, and we found that within the bank, we're consistently getting a good quality product. In addition to that, we've found across banks that we've used, we've had consistently quality products. So that's very important because we're able to pull from a variety of banks and feel confident that we can manufacture the same product using starting material from different places. Timing, again, 4 to 6 weeks for a autologous product to come, where cord bloods, we actually have them already stored on site so that we can manufacture on demand. And of course, logistics, we just request cords from the bank and ship them to us. So this is just a little bit about our manufacturing process. This was the our first process that we had. And this is, we start with a cord blood unit, a single cord, and we wash it according to a process that's very consistent with what most cord blood banks ship out with their products. We then isolate by either, in this case, we enriched our CD25 cells. We cultured those for 14 days, 12 to 16 days. And then from there, we're able to create multiple doses where we cryopreserved and stored for distribution, for later distribution. We have since changed our process to make our different products. So we started with CK001, which was a bone marrow failure clinical trial. That was actually a fresh product since that was one of the first manufacturing that we performed in-house. And we hadn't really qualified our cryopreservation process yet. So we did that with it was a clinical site with one clinical site. We then were going to move on to ALS, but then COVID happened. We had treated a couple COVID patients on a compassionate IND, and we had favorable clinical results. So we decided as a company to shift gears and move to the COVID-19 ARDS indication. So we turned it around. We completed our cryo qualification, and we started planning for that IND. We filed. We included our clinical data from the compassionate IND, and FDA at that point turned around and said, you know, this looks pretty promising. We really would like you to do a double-blinded, randomized placebo study instead of the 9-patient study that you're submitting. So that was a really big challenge. So we had to turn around and figure out how were we gonna label this to keep it blind, how were we going to make our placebo and test it. And so that was a kind of a whirlwind of activity in our area. But we filed because COVID was so rampant. We didn't have any problems enrolling patients. We had 5 clinical sites. So we were able to distribute very quickly and treat our 45 patients from start of our IND to finish treatment in 7 months, which was really a great feat for 12 people. Our third product was actually CK004, and this is to treat myelofibrosis. So again, we switched up some of our manufacturing process to select for a certain particular cell type that we find homes to the marrow. And this is, we formed a collaboration with a company so that we could, again, treat in tandem. So we would use our cells to treat, to kind of calm the inflammation, and then use their drug to come in and treat the patient. So we're about to start that clinical trial in this month, probably end of this month, where we'll start enrolling. And then we have our ALS IND that's currently with the FDA. We've treated 5 patients already on a compassionate INDs. So we're hoping to get through okay and hopefully start our clinical trials soon. And then this was not my slideshow, my true slideshow, but I just wanted to kind of have some take-home points. And really in this forum, it's very important to let you know that what you do really matters. I think maybe 10, 15 years ago, cryopreserving cord blood, you really didn't realize the extent and all these new technologies that are actually using them. So I think by people with high quality and doing what they need to do, it really helps us on our end process and make a good product. Thank you. Okay, so next up, we're going to have Joe Blake. He's currently the Associate Director of Process Development at Devera Therapeutics, where he's been for 2 years. He's been working with expanded cord blood cells since 2006, where he worked with Colleen Delaney at Fred Hutch, where they worked on the expansion of cord blood CD34 cells on Notch ligands. Since then, he's been working with the expansion of cord blood cells, both in process development and manufacturing. So please welcome Joe.
Okay. All right, thank you, Monica. Thank you for having me at the conference. Thank you to everyone who organized the conference for having us. We're all excited to be here to talk about using cord blood. As I said, my name is Joe Blake. I'm the Associate Director of Process Development at Devera, and I'm here to talk to you about our cell therapy expansion platform. We've been working on this platform since the early 2000s. It started at Fred Hutch, and then we spun it out into a company. We use cord blood to generate multiple product cell types to treat different conditions. And our goal is really to have allogeneic, HLA-unmatched cord blood cell products to enable access to more patients with these products. These are my disclosures. I'm an employer. Oh. These are my disclosures. So our current technology uses Notch ligands to expand cord blood cells ex vivo. Notch ligands are found in the bone marrow niche and allow self-renewal of HSCs without and inhibit differentiation. And so we utilize this in the form of an engineered Notch ligand that we use in our cell expansion process that allows us to expand the HSCs many times while retaining their HSC function. And in doing this, we've created a standardized cell therapy manufacturing process. And then we can further take those cells and differentiate them into different cell types. And so that's what I'm going to talk to you about today. So our cell therapy manufacturing process we have standardized, and it consists of a number of steps. So we take cord blood units and we isolate the CD34 positive HSCs on the Clinimax, which some of you may be familiar with, and then we expand them, as I said, on Notch ligand. And in this Notch-mediated expansion, we get a good expansion of the HSCs while retaining their HSC function. And then we come to sort of an inflection point. So we can either utilize those cells, which we have done over the years in as expanded HSCs as a product by itself. That product has the generic name Dilanubicel, so we'll be talking about that. I know that's a mouthful, but I can teach you to say it later. Or we can differentiate them into NK cells, either modified or unmodified, or myeloid cells, or other cell types. And after we do either that differentiation, we put them into our standardized fill-finish process. Cells are cryopreserved, ready for off-the-shelf use. They can be released to be used for any indication we choose. So, as I said, we start with cord blood cells. So cord blood is a highly regulated product. I mean, it has a lot of advantages that way. The NetCord FACT people do a great job of standardizing the process and helping banks get a standardized process. And today, about 20% of the available US inventory is actually licensed, and it's regulated by the FDA. And so when you go to the FDA with a cord blood product, they know what to expect from your starting material, which is advantageous. As well, the storage of cord blood has been validated. This citation I have here is from Hal Broxmeyer from 2011, but if you saw James Ropa earlier today showed they now have data that's out 27 years of storing cord blood and still maintains its HSC function, which is, great to see that they're continuing Hal's work and that, we're really validating that we can store cord blood presumably indefinitely and still use it and have good functional cells from it. It also has biological advantages, as Monica mentioned, and so it requires less matching, which we know from cord blood transplant. There's a lower degree of matching required to achieve engraftment, although some people in some of the other sessions may have debated that a little bit, but they, it's easier to get a cord blood unit to engraft with a lower degree of matching versus a peripheral blood stem cell source. And what we've noticed is that in our Notch platform, we've grown HSCs from bone marrow, cord blood, and peripheral blood, and the cord blood cells rather grow much better. And we think that's due to them being a much younger source of stem cells, and they seem to have greater expansion capacity on Notch. But the biggest limitation of cord blood from a technical perspective is the number of cells. I mean, the average cord blood unit is less than 10 million cells. I would say it's probably more like 4 or 5, but when you look at a starting cell source, if you're using HSCs like CD34s and you have 10 million of them, you can only make a couple of expanded products from that. And that will raise your cost of goods in any manufacturing process. And then the other disadvantage or function of cord blood is there is donor-to-donor variability. I mean, cord blood banks have release assays that assess CD34 and colony-forming unit content, but we know when we put them in our expansion process, they have variability in growth. So to overcome this, and there's a long history of this technology being developed, we initially developed this process at the Fred Hutch, expanding single cord blood units, and we eventually switched to pooling cord blood. So we take a number of cord blood units, pool them together, and then run the enrichment process and then the expansion process, and then cryopreserve the resulting cells as our cell product. And in doing this, we've made a much more consistent product, and we've allowed larger batch sizes. So instead of 1 or 2 or 3 products from a single expansion, we can get 5 or 10 or 20. And I'm not just, so I'm going to show you some data. I'm not making this up. So like I said, this goes back to the early mid-2000s, and so we have expansion process from our HSC product, Delanubicel. We have expansion data from approximately 150 single donor cord blood units that we enriched on the Clinimax, put in our Delta expansion process, and cryopreserved the resulting cells as a product. We also have about 50 lots of pooled expanded cells. And those cells, for this data, it's all 8 cord blood units that were pooled together, enriched, and then expanded. And so I'm going to show you the data on the performance of these cells in our expansion process. And the goal of this is to increase the total number of cells we have at the end without compromising cell purity or expansion performance or yield. So I will mention that I did, back in 2012, write a paper on using the Clinimax to expand cord blood, and that's the process that fed into this development of this pooled process program. So the first thing to note here is the first graph on the left is the total number of CD34 cells we enriched from each cord blood unit, and as you can see, the vast majority are well under 10 million total cells. And on the right in this graph, we have how many cells we enriched from pools of 8 cord blood units. This is a little biased in that we picked the number of cells we put into the system. So some of these we may have wanted to get more out, and some we may have wanted to need fewer, so we could have picked smaller cords. But all in all, we tended to average putting about 30 million cord blood CD34 cells into the system, and this is how many CD34s we get out. The second graph shows the post-enrichment CD34 percentage. As you can see, there's really no difference between single or pooled donor cords. And then the third graph is the efficiency of the selection process. So one of the things we were concerned about is if we loaded a whole bunch more cells in, would we recover 10% instead of 50 or 60%, which is about typical with the Clinimax. But it's very consistent, and these numbers are almost exactly the same. So we were really encouraged by this data showing that we can put more cells in, pooling cord bloods together. We're not losing any purity or enrichment efficiency. And you can see that there was quite a bit of variability in some of these single cord units. But this was data we generated in our lab, and this is all based on data provided to us by the bank, which is great because you get your product, you don't have to test it, you just put it right into the process, and the banks do a very good job of having reliable data. So the other thing I want to mention is this data represents about 550 cord blood units in total, so about 400 units in the pooled and about 150 in the non-pooled. And so we selected our cells. Now we've put them into the expansion system. So we wanted to make sure our expansion system was working correctly and not impeded by the pooling. And what we found is that we get about 1,000-fold expansion of CD34 of total cells from pooled CD34 cells and single cord blood. But look at the variability here in single cord blood units. I mean, some of these are expanding 10 or 30-fold, and one expanded more than 3,000. But in the pooled group, we've managed to shrink that variability quite a bit, which makes it much more consistent. And likewise, the post-culture CD34 purity is a little bit higher and more consistent in our pooled culture process. This probably looks low to people who are used to seeing, enriched CD34 cells, but the HSCs that remain here, the CD34s, are still multi-lineage engraftment, and are multi-lineage, give rise to multi-lineage colonies and gems in the colony assay, and they have expanded 200 or 300 times. So you have a huge quantity more CD34s, even though they're not quite as pure. And then, the whole point of this endeavor was to get more cells out of our system at the end. And it's notable, this is a log scale. So by pooling 8 cord blood units together, I've gotten consistently about a log higher, really about 8-fold higher, which should make sense, output of my system. And again, we've shrunk the variability down in the output here. So we all in all have a much more reliable expansion process. So one of the things to think about when you're manufacturing a cell therapy is your cost of goods. That's going to drive, at least in the US, it's going to drive how well you can make this a commercial product. And by starting with cord blood, you have this huge variability. It limits your ability to predict your lot size. It limits your ability to schedule, and it makes it more difficult to predict how much it's going to cost. So by pooling, we've managed to streamline this process both by reducing the costs associated with manufacturing, hours in plant, number of runs to achieve the same amount of product, and when you're releasing a product, you have to do release assays on it. Those can be somewhat expensive, and if you have to do one for every single bag, it's extremely expensive versus if you can do one for a lot size of 40 bags, that's quite a bit of savings right there. And then it also, because we've increased the consistency, we've made our scheduling easier, our lot sizes more predictable, we're more able to make demand for our clinical trials. So, as I said before, we utilize notch expansion for expanding HSCs, but what I did mention is that we can also take those HSCs and produce different cell types and a number of variety of cell types from these HSCs that have retained their function. So now I'm going to show you data on, well, not a lot of data, but some, but I'm going to tell you about our NK cell product, which we take the same Dillon cell process that I told you about. We pool cord bloods, enrich them, put them in the notch expansion. After that notch expansion, that ends up comprising the first phase of our NK cell manufacturing. And then we put those cells directly into an NK cell differentiation process. And what we found is that we can make allogeneic NK cells, we can make large numbers of them, and it just feeds from one technology to the other. And what we found is that the notch expansion process actually is important to creating these NK cells, because if you do that two-step culture process, but you don't use notch at all, you get relatively few resulting CD56 NK cells out of the NK cell process. But if we use increasing densities of our notch ligand in our culture system, we end up generating large percentages and large numbers of these CD56 positive NK cells. And so we have optimized this process to produce our NK cell products. And it is worth noting, some people take NK cells, pull the NK cells out of the cord blood. These are not cord blood NK cells. These are de novo NK cells derived from CD34 HSCs, because the selection process eliminates the NK cells. And so we've been able to generate this very consistently. And so you can see here the percentage of CD56 cells generated in culture over the expansion phase is very, very low. But once we put them in our NK differentiation phase, we rapidly convert them to and generate CD56 positive NK cells. And we're getting a 2,000 to 2,500 fold expansion of these NK cells. And then this graph is a cell killing assay showing that these cells have function. I'm not showing you, our cells do express a number of canonical NK cell markers such as NKp46 and CD16, but I'm not showing that here, but we've assessed their function against a number of tumor cell targets, including this A549 target cells. And the way to read this is we've co-cultured our NK cells with tumor cells at increasing effector cell to tumor cell ratios on the x-axis, and then we've measured the cytotoxic effect on the y-axis. And our cells outperform, in this assay, outperformed the cell killing mediated by adult-derived NK cells, which we took as a control from normal peripheral blood. And we've done this against a number of different cell lines. Some are resistant, but our cells do kill a number of them, including the canonical K562 line. And so now that we have this platform, it is very amenable to lending itself to genetic modification. So if you want to make a genetic modification, say inserting a CAR as an effector function, or knocking in or knocking out a gene to increase a cell's fitness in vivo, there's multiple points in the cell process, this cell culture process, this could happen. For instance, if you wanted to, very early on, you could transduce the CD34 cells to express a CAR, assuming the CAR target isn't expressed by your cells throughout the process, and then you would need a very limited amount of say, retrovirus or lentivirus to do that at the beginning and generate a large number of cells. Likewise, if you wanted to transduce late, or if you were transducing with something that would be somehow inhibit the expansion process, then you could transduce late, and there are various technologies to do that that we're investigating. And of course, you know, with the COVID vaccines, everything LNPs are quite popular right now. So now I've told you about our HSC product that we started with and established our pooling protocol, and I've told you about our NK cell product, which we derived from our HSC product. But as I mentioned, we have developed a technology, a process to create monocytes and macrophages from our expanded Dillon cell product as well. So we've taken the same sort of methods to beginning to expand, and then we can generate monocytes, and we can further differentiate those into functional macrophages. And you know, there's multiple opportunities at when you might want to use those or cryopreserve those depending on your disease indication. And if you want to know more about that, we do have a poster. My colleague Shannon Kreutzer from Devera authored, and it'll be at the poster session. So I'm not going to talk about that today, but that is out there. So in conclusion, our notch ligand process allows us to expand cord blood CD34 cells, and we've really utilized pooling of cord blood cells rather than a single unit because it's just not economically feasible for us in the long term to use single units because of the cost of goods. And we can utilize those cells to make very potent NK cells. And this platform lends itself to genetic modification in a number of different steps. And we can create cells other than NK cells, which again could be modified in any number of ways. So this is, there we go. I'd like to thank my colleagues at Devera, and in particular, I highlighted Colleen. She'll be moderating a session tomorrow, and then Devika is giving an oral abstract on the CAR NK cell therapies, if you want to hear more about that, tomorrow at 5. All right, this is jumping around on me, sorry. And then we do have 3 posters available. I'm not going to touch anything now. 3 posters available if you want to look at that. I'd also like to thank, as I said, this work historically came out of the Fred Hutch, and so I'd like to thank our colleagues there. We still have ongoing collaborations with them. And I'd like to thank the Cleveland Cord Blood Center, who are here today, and we've worked with them to source cord bloods, and that's all. Thank you.
Okay, so our last presentation is going to be virtual, and it's going to be given by John Wagner. John Wagner is a director of the Institution of Cell, Gene, and Immunotherapies at the University of Minnesota. As many in the audience will know, he performed the first transplant for leukemia in 1990 and pioneered the use of the double cord transplantation in adults in 1999. The topic of his presentation today, however, is not going to be on transplant, but rather on the benefits and challenges with cord blood cells as starting material for deriving induced pluripotential stem cells.
Good afternoon. Thank you, everyone, for the kind invitation. Unfortunately, I couldn't be with you in Miami this afternoon due to a concurrent conference in Texas. Certainly, if anyone of you has a question about the presentation on cord blood as a source material for novel iPSC-derived immunotherapeutics, please email me at the address shown below on the screen. I have no relevant disclosures for this presentation, although I work with a number of biotech companies. None of these relate to this particular presentation. In 2007, the proof of concept that iPSCs could really have therapeutic value was reported in a mouse model of sickle cell anemia. In 2014, Takahashi in Japan initiated the first clinical trial with iPS-derived retinal epithelial cells with promising results in patients with wet macular degeneration. In this first study, the iPSCs were generated from dermal fibroblasts from a 4-millimeter skin biopsy. But since that time, iPSCs have been generated from various somatic cell sources and reprogrammed with integrating viral vectors or integration-free systems. In general, there are the 3 steps as shown here in being able to make an iPS clone cell line available to us. While not the specific focus of this particular presentation, we certainly need to keep in mind that iPSCs can play an important role in drug discovery, as well as cell or tissue degeneration. As shown, the development of iPSCs from patients with a genetic disease, for example, can be very useful for high-throughput screening of compounds that could modify or reverse the disease phenotypes. Examples of diseases for which this approach appears promising is shown on the left-hand side of the panel. For example, in ADA deficiency, muscular dystrophies, Parkinson's, and so forth. So one question is, what is the best starting material? As I said, reprogramming of any somatic cells is theoretically possible. However, certain subpopulations are more amenable to cellular reprogramming. Adult stem cell populations such as those from muscle, brain, liver, and the hematopoietic system appear to be superior to cellular reprogramming compared to their differentiated counterparts. Another factor is epigenetic memory. While pre-programming itself erases most of the DNA methylation patterns of the starting cell, low passages of iPSC harbor particular methylation patterns characteristic of the cell of origin, impacting upon the overall success of iPSC derivation and its usefulness. Also, cell age-dependent differences in differentiation capacity, as well as DNA damage repair mechanisms, also play an important role in reprogramming efficiency. Therefore, we wanted to focus on a cell population that is highly efficient to reprogramming, and ideally would be obtained from a readily available younger donor. And for these reasons, we then thought of neonatal cord blood or placental tissue potentially offering some important advantages. Now, in terms of the advantages or disadvantages of an autologous versus allogeneic stem cell source, you obviously know much of this already, but just briefly, autologous products would be less likely to elicit an immune response and less risk for the recipient of new pathogen exposure. But unfortunately, the cost of making cGMP grade autologous iPSCs would be exponentially greater because of its use in a single recipient as compared to an allogeneic off-the-shelf product which could be used for many different people. Of course, that's presuming that we have strategies for limiting the risk of rejection. So therefore, that's a major area of focus of what we like to discuss today, but also to think about, there are other optimizing engineering steps that would be tailored to the individual product for which we are interested in. And we'll talk about that in a second as well. But in terms of how to deal with the HLA matching issue and its effect on rejection, we know that HLA match is important, particularly so in the allogeneic hematopoietic stem cell transplant setting. We also know that rejection is also a barrier to successful solid organ transplants in the long term, and it requires lifelong immune suppression in most cases. But to diminish the impact of HLA, there are a number of strategies beyond prolonged immune suppression. For example, we can identify donors with common HLA haplotypes as one approach. We could also co-infuse tolerizing agents like regulatory T cells. Alternatively, we could knock out HLA loci, and there are other strategies as well. But the idea of identifying the most commonly shared HLA haplotypes has already been considered. And this is just one example. This is it for South Korea, where there's less HLA diversity, somewhat like Japan. And it's been demonstrated in this particular study that 48 donors would be all that would be needed to have an HLA-matched product for nearly 80% of the entire population. What you can also see here is that it only takes 11 to 12 donors for covering 50% of the population. So in my mind, this certainly begs the question about, can we demonstrate the proof of concept with just these 11 to 12 donors, that this is the most cost-effective strategy with lower risk of rejection. But HLA genes are closely linked and inherited as haplotypes, as you all know, with one haplotype coming from mother and one from father. So this is somewhat limiting in our ability to find the kinds of donors that we want. Now, the one thing I want to point out to you is that, we already know that many of the haplotype frequencies in the United States, and we have it broken down by race and ethnicity. And certainly, we know what the common haplotypes are with each of these different populations. And there's sharing of haplotypes between the populations as well. However, this may not be the only strategy for identifying, a small number of cord blood units that could be useful in the types of cell products we're interested in. So for example, with cord blood Tregs, we previously showed that partially matched to 3 alleles was sufficient for preventing graft versus host disease in the setting of a double cord blood transplant, where the risk of grade 2 to 4 GVHD was at 62%. We demonstrated that only one patient in that study, that is 12%, developed a GVHD-like reaction, suggesting that cord blood Tregs don't need to be matched at all alleles for it to be an effective stem cell, effective immune modulating strategy. Because making individualized products in that particular study took so much time and, more importantly, was so costly, we wanted to ask the question whether or not we could identify a small number of units that can provide a 3 allele match for most of the patients. But if not that, we wanted to at least have one match to see whether or not that would be of equal potency. So we then asked the question, is there a combination of desirable HLA alleles that permits some level of matching with nearly everyone, regardless of race and ethnicity? The primary aim was to determine how many donors would be needed to ensure one allele matched at A, B, C, and DRB1, which is a very different question than how many donors would be needed to ensure complete matching. Furthermore, of these small number of donors, how many would match more than one allele? The prediction was that matching one allele would probably take still a fair number of donors across race and ethnicities, but of those, probably very few would match at more than 1 or 2 alleles. Well, we were surprised. In an analysis of these 7 cord blood units that were identified that would match nearly everyone based on one allele, considering A, B, C, and DRB1, we were surprised that when investigating how many of these 7 cord blood units would provide at least a partial match at more than one allele, we demonstrated that 80% of the searches that were taking place at that particular time of an NMDP that would match at 3 or more alleles. So at least one, in this context of making a bank of Tregs from 7 donors, looked very optimistic. So I'm not going to go into detail about how these 7 cord blood units were identified, but nonetheless, there were 7 cord blood units that were predicted to match nearly everyone, and it did so in 99.95% when looking at an active search group of patients searching for an allograft. And 79.2% of the time, they matched to 3 or more, and you can see the distribution by 2, 3, 4, 5, and 6 allele matches. But this turned out to be more challenging. Despite the fact that we thought this would be a great way of identifying a way of having 3 or more matched Treg products for future clinical trials, this demonstrated a few obstacles. One is that cord blood units were found at multiple cord blood banks. That is, these 7 units were not all in one bank, as you might expect, but this also means the need for multiple agreements. This by itself was not a major barrier. The second, though, was the consents used for the original collection of the cord blood unit. These varied over time, and none were adequate for the work that we wanted to do, that is to make potentially a clinical grade product and potentially of commercial value. So this meant creating new documents, getting buy-in from the cord blood bank, as well as the University of Minnesota General Counsel's Office, and then reconsenting, with the latter being actually the biggest challenge, perhaps. Lastly, the desired HLA combinations were sometimes rare, and only one or no backups were available. So we had to consider that this source was going to be limited, and this is what led us down the path of considering developing iPS cells and then genetically engineering them the way that we would like to. So we developed collaborations between experts in stem cell programming, that is within the Stem Cell Institute, gene editing, that is in the Cancer Cardiovascular Research Center, immunology in the Masonic Cancer Center, and product development / GMP manufacturing at our MCT facility. We identified the needs of the faculty as a whole with a particular interest in the development of iPSC-derived therapeutics, and our particular interest in my group was in cancer immune effector cells in the Cancer Center, but also there were interests in deriving muscle keratinocytes, beta cells, as you all would expect. Faculty in these centers and institutes would provide broad expertise in different areas of genetic engineering and synthetic biology. For example, there are potential effector cell enhancements like gate switches to ensure more precise targeting, that would in turn eliminate on-target off-tumor side effects, suicide genes to ensure safety, survival enhancements such as the editing of CCR5 gene for HIV therapies, or providing enhancements that improve function and persistence, such as with regulatable CARs, addition of surface-kind cytokines, and so forth. Interestingly, the development of mutated receptors that respond to orthogonal cytokines is also a particularly useful approach for selective stimulation of the iPS-derived immune effector cell population of interest. So the best starting material was also under investigation, and so we wanted to be able to take a cord blood unit and select out various subpopulations as shown here as to be used as the starting material from that cord blood unit. And so this has been worked out as well. The aim is to use non-integrating strategies and focus on the development of allogeneic off-the-shelf products as previously stated. And our particular interest in my group is in developing immune effector cell therapies, new strategies that eliminate the need for lymphodepleting chemotherapies and enhance more precise targeting and killing, which is all under investigation. So these are just giving you some additional ideas on what we're moving forward to be able to move these immune cell therapies in the context of cancer. While the NCI now sees cell therapy as the fourth pillar toward cancer cure beyond surgery, radiation, and chemotherapy, we're asking the question whether or not in the not-too-distant future antibodies and off-the-shelf immunotherapeutics could become first-line therapy, potentially eliminating chemotherapy for some diseases altogether. The overview of our plan is shown here for cancer immune cell effector cells. What you can see is that we first do a CD34 purification, for example, as our starting material. Then we do the reprogramming, additional engineering steps such as the B2M knockout, so removing HLA class 1, and full characterization of the population, followed by cryopreservation. Now, this is the population we plan to make available to all investigators, but in our own specific projects, we will be introducing a CAR, followed by additional engineering steps to enhance persistence or more targeted expansion possibilities, and then developing protocols for differentiating them into T cells, whether it be alpha beta or gamma delta T cells, Treg populations, as well as thymic progenitors and NK cells. So certainly this is the work of many individuals at the University of Minnesota. And as you saw the slide before, I'm now giving you the names of the people: James Dutton and Brenda Oglethorpe at the Stem Cell Institute, leading the effort in reprogramming, Bo Weber and Brandon Moriarty at the Cancer Cardiovascular Research Building in charge of the editing, Darren Sumstead and David McKenna at the MCT facility, and then the group that I work with, Bruce Blazer, Jeff Miller, and myself, who are focused on developing immune effector cells. Thank you for your attention, and I'd be happy to address any questions you might have if you send me an email. Thanks so much.
Okay, so we're going to open it up now to questions, if anybody has any.
I have a question for Joe. So first of all, I want to know how you get your NK cells. So because usually if you can isolate CD34, and then from the CD34 you put SCF, FLAT3, IL-7, and then IL-15 in it to get NK cells. So is your first step of expansion together with SCF, FLAT3, and IL-7, and then you add 15?
So the NK cell expansion process is somewhat proprietary, so I can't go into great detail. But what I will say is that, as I alluded to, we really use the first step is essentially the Dillon-Ubicell process, which we've published on before. So we do use SCF, TPO, and then we transition them into a new culture condition.
And then you get, if you culture them first for 7 days or 10 days or whatever, and then how is your percentage of NK cells you get out of your, at the percentage, CD56, for instance?
So it varies anywhere from 75 to 85%. There are some, both our HSC product and the NK cell product has some myelomonocytic type cells in there. Again, no, we don't see any T cell development.
Okay. And so what is your, why do you could also do this in parallel to expand hematopoietic cells on one side and then go directly to NK cells, for instance?
So you're asking why don't we do both?
Yeah, to parallel. Yeah,
I guess the complexity of manufacturing at this point, we've never thought about like trying to make a combination. If I think I understand what you're asking, we haven't really tried to make, I mean, we're not trying to achieve long-term engraftment with these cells either. So, in the interest of a single cord blood, you might say we would make some NK cells and then some expanded 34s and put them all back together.
No, my question is more, if you compare, for instance, you can, because we have an NK cell group at our institution, it's not my group, but what they usually do, they do a CD34 and then they expand them for 1-2 weeks as with SF3 and maybe IL-7, and then they get a huge expansion already of CD34 and then they add the NK cell specific factors. So,
yeah, I think that's what we're doing. Yeah.
Okay.
Nice talks, Joe. Is there any criteria for the cord blood units that you pool? Do they have to have common ABO types or?
No. So there are criteria. I mean, first and foremost, in a clinical setting, they have to be qualified for use in transplant, essentially. And then the only criteria we have set, we're agnostic to HLA or ABO or SACS or any of that. The only criteria we've set is number of CD34s, and that's really, I mean, we set that fairly low, but, they have to have at least, I think, around 2 million. To really, at this point, you could imagine a scenario if we were pooling 40 cords, we could take smaller cords, but there really isn't any. Our strategy has been to ignore the HLA, ABO types.
That leaves the inventory wide open, doesn't it? Thank you.
Yes.
Joe, I have a question too on the pooling, because I think that's very interesting and something that we would like to kind of move forward with. We're working on. How did you go about the process? Did you start with, let's start with 2, let's start with 4, or did you go jump to 8?
So this all goes back to when we were originally starting out, back in the early 2000s and maybe even the late 90s, where we commonly pooled CD34, cord blood CD34 to expand them in our initial discovery research. So we knew it would work in terms of the expansion. And so then we ended up with 8 in this setting is more of a technical, like logistical capacity. We've done as many as 12. There's no reason to think the system couldn't handle more than that. And we've done as few as 4 or 6, but we wanted enough cords to have that, to reduce that biological variability that we see in expansion. So if one isn't going to expand very well, if you do 3, then that's a third. But if you do 8, that's what, 1/8. So
and do you wash them together, or you pull them together before you before you select them?
Correct.
And then, okay. And you haven't seen any issues with that as far as?
No, the issues we see are related to the red cell content of the frozen cords. There tends to be some, in the processes for cryopreserved cords as they're frozen with the CPAX or the AXP, there's still a large hematocrit in there, and so that, we're trying to get rid of that, and so we've had a number of issues that we've had to mitigate there, so that's been the biggest issue.
No viability issues combining them?
No, and we're a little privileged in that the HSCs we get out are always very viable, so if there's some other junk cells in there, they just go away in our expansion system.
Okay, thanks.
So I think to kind of extend the discussion on the pooled, and depending on what's proprietary versus not proprietary and what ends up being maybe like service burden, to have to do it. One of the things to maybe keep in mind, especially if other groups have interest, is if there's room for, let's say, a characterization or a method process around it, especially if it is a pain for you to have to do it, yourselves, and this might have been what piqued Donna's interest, is if then there is sort of like room to take it off of like an individual group's plate to have to do it on their own, and if there's like a wider room for like a service provider, out there to like a HemaCare type of group or any of those Key Biologics, and their new names and everything like that, Charles River. And so that might be something where either a commercial group or even maybe an NMDP or something like that might then be able to say, if there are 12 characteristics, that we're looking for, or things you definitely don't want to have in there as far as the pooled or the testing that might go through, then maybe there's like almost a template, that can go through, and then someone else might be able to then say, instead of 5 groups pooling it on their own, having to put in your own service folks, labor burden, it might be something that someone else might be able to aggregate and actually then in the long term make more efficient. If it ends up anything I said, actually makes any logical sense, it might be something we want to think about for a topic for like next meetings or something like that, and see if we want to bring in someone who might be interested in actually doing. Yes, yeah, and so they turn around and they sell it to you, and hopefully in the long run, they're making money off of it, but then you're also saving money by not having to do all of it yourselves and having some, let's say, GMP characterization type of thing. Yeah, and so again, the individual banks, and so that's an opportunity, but then if, let's say, you have to go to 6 banks to get enough for what you might need, it might not make enough business sense for each bank to have to invest in it on their own, but then someone else, but then there might be an alliance or an NMDP or something like that, So obviously there are some wheels turning going on as it was said in real time, but it might be something that we pull on that thread for a conversation and maybe keep in the back of our heads if it's something to kind of bring up as a session next year or something, so.
So I think that's a really good point, and as I mentioned with Monica's question, the biggest challenge around dealing with the cord bloods is how they're packaged, that's the biggest technical challenge, hurdle that we deal with in the actual manufacturing environment. So decoupling the pooling and enrichment potentially from from our manufacturing process would be, potentially very viable. It comes with a number of regulatory issues, because then you're talking about a bank or another group supplying, instead of a minimally manipulated product, a more than minimally manipulated product potentially, and viewing it as a starting material in a different way, which might make the FDA throw you some curveballs. And then the other challenge is getting enough cords in. I guess there is one I didn't mention, the starting material criteria we do have. We do need the cords to be frozen within 48 hours of collection, which is pretty common. But I just want to point out that if you're going to pool them fresh, you need to collect a bunch of cords right away, pool them, and do that before you freeze them within that 48 hours. Unless you're thinking about, because then if you take your, so say you were going to take your existing inventory, thaw it out, enrich it, and then freeze it again, you're going to get another hit from the freeze-thaw, double freeze-thaw, and you're going to lose a lot more cells, so that's not going to be cost-efficient either. So I think as a more progressive, moving forward sort of idea, it's a really attractive solution, but I don't know how using existing inventory gets us to that place.
Yeah, and what it feeds into is kind of this whole theme of platform technologies. Mm-hmm. And so basically, you got, 10 groups who do 1 or 2 cords at a time, and that tends to not be worth it necessarily to invest, and sometimes not even have enough fluency on it if you're not working on enough. But then that's where you might either have like a, pooled resources, to kind of play on words. But then the other aspect to, before I forget, you're mentioning then sort of like you're starting with something that's not minimally manipulated, has its own regulatory footprint, let's focus on that. Then you move to something that you're doing something, let's call it a manufacturing step. You're making a product, basically, and you're characterizing it. It's going to have a certificate of analysis. It doesn't necessarily need to be regulated. It could be a starting material for what someone's going to use it for, and then if you do a master file with the FDA, that then turns out to probably satisfy the checkbox for what then might, because now you're putting it in that position of a raw material and not necessarily something that you're saying that pooled set of cells is quote unquote regulated, because whoever's selling it or doing the service providing is not saying we're going to use it directly as a clinical, therapeutic product or as a device or something like that. It's a raw material. So there's a pathway for it, and probably just talking off the top of my head, which actually might be off the other end of myself, but it's really, I think, actually a master file for something like that, if it's characterized, would actually be the simplest way, and then anybody who uses it can cross-reference that. So
yeah, I think the other thing to think about, though, is to not shoehorn ourselves. We have cord blood, and our group uses the CD34 HSCs. There's other cell types in there that someone might want to use at some point, so we don't want to throw away that negative fraction, but unless we know what the demand is. So if someone wants the monocytes, that's great, we can take them out and freeze them, but we have to know what to do before we freeze them. So shoehorning yourself in to just doing the CD34s, and so I guess that's my perspective as someone who started in the lab, getting the cords that were anonymously donated and using them, and they were a very precious resource. So I don't want to have us throw away part of something that could be used someday, but at the same time, we have to make it cost effective. But that's just another thing to consider.
You're totally right. And, if I were hypothetically a manufacturing or business person, what you could end up doing is you're taking that starting material, let's say the cord blood, you might be, processing it to isolate the different cell types. Each of those is its own product. And then someone could pick and choose, almost saying, if I want to put together a CD34 with a monocyte or something like that, here's catalog number 1 and catalog number 2. Each of them are characterized, and then, they might end up feeding that into, their own needs as opposed to another company's needs, that might be different. So there seems to be some potential pathways, but right now it may not be efficient enough, and that's why you probably have to do a lot of the work yourself. Just in case, Joe, you want to do less work. If you want to, keep doing more work, no problem. But so anyway, just some thoughts and ideas, and maybe something we can, discuss, as we start tomorrow, probably, or 3 days from now, planning for next year's, conference. So,
thank you.
Okay, I think that concludes the session.